So we'll continue our look at humoral immunity by looking at antibodies. Now, antibodies are often called immunoglobulins. They're globular proteins that are found in the blood plasma. It's one of the three proteins that we talked about in blood plasma. And they're sometimes called gamma globulin. So these are proteins, and they're secreted by the plasma cells, which are the activated B lymphocytes that are grown in the plasma reticulum. And their job is to bind with a specific antigen. The specific antibody binds with the specific antigen at the antigenic determinant. And so they are secreted by the B cell, plasma cell, that has that receptor to that antigen on it. So they are very specific. They kind of are a Y-shaped thing of polypeptides, peptide bonds, so they're amino acids joined together in a chain. There's heavy chains and light chains and all kinds of things that really don't help you understand how they work. So we're not going to spend any amount of time on it. But basically, there's 5 different classes of antibodies. And they're not 5 different types, but classes. We'll just leave it at that. And it spells MADGE, M-A-D-G-E. There's IgM, IgG, IgA, IgD, IgE. MADGE is the mnemonic to remember it. And each class of antibody has a different job to do in helping eliminate the antigen, which is the ultimate goal of all of the adaptive immunity. So they end up looking like this, and each antibody has got a couple of large proteins, a couple of small proteins. There's 4 proteins, and they're kind of hinged together, and there's antigen binding sites. And this is the only part that really matters, is that right here at the end of the Y's is the part that actually binds with the specific antigen. This part is the part that's variable from antibody to antibody within each class. So the IgMs is kind of a star-shaped thing. It looks a bit like a snowflake. And what it does is it agglutinates. It joins things together. It's kind of like a chain gang. Well, I'll explain it more in a minute or two. But it's an agglutinating agent. It also is involved with activating complement. It targets these pathogens for complement. Now, IgA is a simple one that that is found in mucus and other secretions outside of the body, and it binds on antigens and on pathogens and prevents them from accessing the body very easily. So IgMs are these, and you'll notice that they're kind of like I said, a snowflake. So in this case, there would be 10 antigen binding sites. This one is got 4. It's kind of 2 joined together, as it were, with 4 binding sites. This one acts kind of as a chain gang, and this one acts more like handcuffs or leg irons. And we find these ones in all kinds of secretions. IgD is a monomer, so there's only one Y-shaped thing, and it is the B cell receptor. It functions as the receptor to the antigen on the surface of the B cell. IgGs are the ones that are the most common. Most of them are, like there's 85% of the antibodies in your plasma are these. These are the ones that can cross the placental barrier. These are the ones that that we use to give passive immunity. These are the most potent ones. IgEs are involved with mast cells and basophils releasing histamine. So these ones are actually involved in inflammation and causing inflammation. So they end up looking like this, and you'll notice that all three of them are monomers. So they have the two binding sites and the one base. And kind of go from there. Each of these things can be produced by the B lymphocyte. I'm not going to worry about most of it because most of it really is beyond what we technically need for this course. It's suffice it that there is a specific antibody or antigen binding site that's unique to each B lymphocyte, and it's unique for each antigen, but it can come in all the classes, and that the plasma cells are capable of producing any of the classes here. So, but this is more important. What do they do? So they inactivate and they tag the antigens. They actually, by binding to the antigen, identify the pathogen or the thing that has the antigens on them. Now, it's useful here because it causes either neutralization or agglutination or precipitation or complement fixation. All of these things happen. Start out with neutralization. Neutralization is when you've got, so say you've got a pathogen, a bacteria that's in your interstitial fluid, and it's covered with proteins, you know, in the cell membrane, and every one of these proteins gets an antibody kind of stuck to it. Pretty soon that pathogen is completely coated in these antibodies, and then the pathogen can't do anything. It can't swim. It can't infect anything. It's completely neutralized. It's not dead, but it's neutralized, right? It can't get to the receptors on tissue cells, right? And it becomes the place that the macrophages grab hold of to undergo phagocytosis. It means that these pathogens can't get away and are more prone to be phagocytized. So neutralization is a very good thing. It's kind of like a swarming. Now, agglutination is similar, but it's different in a fundamental way. It's linking more than one pathogen together. I've made mention already of a chain gang. Especially in the southern states, what they do is they take prisoners to work out on the side of the road, clearing brush and that kind of thing. And what they do is they put a cuff on each of their ankles, and then they put each cuff to a long chain. And the idea being that one guy can't run away because they all have to coordinate and run together if they're going to get away. And then the chain is going to trip them up and make it difficult for them to get away, so the cops can get them. Well, this is the same sort of thing. If you take one of these star-shaped or snowflake-shaped antibodies, and you stick it to a number of different pathogens at the same time, the antigens on a number of different pathogens at the same time, you basically are going to clump the pathogens together. That's called agglutination, gluing them together, as it were, and make them not be able to get away from the macrophages or whatever's going to eat them. Ultimately, it's the macrophages that will eat them. So it's basically a clumping it together so that they can't get away. Now, precipitation is the same sort of thing, except what it does is it makes the molecules too big to be soluble anymore. And they end up precipitating out and becoming kind of like a dust, a suspension, and it makes them more susceptible to phagocytosis. They can't get away because they're not dissolved in the plasma anymore. Now, back when we talked about complement, we said that complement was a non-specific thing that was involved in cellular lysis, poking a hole with the membrane attack complex, and with opsonization, making things, giving a handhold for the macrophages to eat the pathogen. So it pokes holes in the pathogen and it makes it so that they can eat the pathogen, and it enhances inflammation. It starts that inflammatory response. And really, how do we know where we target this complement? Well, the antibodies give the target for the complement. So really, this is the main thing that happens against bacteria. Any sort of cellular pathogen gets dealt with through this methodology. Again, we said this in the non-specific when we first talked about complement, but we'll say it again, and it's a list of 3 things. It amplifies the inflammatory response, it enhances phagocytosis through opsonization, and it pokes holes, cellular lysis. So these are the things that happen with antibodies. Antigens meet antibodies, they join together. This causes neutralization, agglutination, and precipitation, which enhances phagocytosis. It fixes and activates complement, which leads to membrane attack complex, cellular lysis, inflammation, which is involved with chemotaxis and keeping things in one spot, and it enhances phagocytosis. And you'll notice that all 4 things that the antigen-antibody complexes do, so what antibodies do, is enhances phagocytosis. Antibodies don't kill anything. Antibodies make it easier for the macrophages to kill them. I'm not worried about monoclonal antibodies. These are commercially prepared, pure antibodies, and what they do is, this is a way of trying to give passive immunity in a very strict, kind of almost at an industrial scale. It's made the development of serums so much better.